Okay. So from the other day, one of the things we mentioned was what was like the major point from the other day? Okay. So well, cells need ATP. I think that's one major point. And when they do respiration, about how many ATP could they possibly be producing? About 37 is a grand total. Now the numbers are different depending on like which textbook you're reading, because and really it's actually dependent upon the individual. But I would say that there's a potential to yield on average between 37 and 39, so I'll just split the difference and say 38. That's 38 ATP. And that's if the cell is aerobic. And though oxygen is everywhere inside of a lot of cells, it's not inside of each cell. There are plenty of prokaryotes that actually are poisoned by oxygen. What we would say, we would call them obligate anaerobes. But this aerobic requirement to generate all this huge amount of energy means that we've got to be talking about being inside the mitochondria. I asked someone earlier, I said, what does aerobic mean? And they said, the 1980s. And I said, no. And then someone said, Richard Simmons. And I still replied, no. Though they both are making reference to aerobics, which is a very popular trend in the 80s, the term aerobic literally means to use oxygen. So aerobic means that there is the usage of oxygen. Now you'll notice I didn't write O, I wrote O2. Hopefully you remember that oxygen is a diatomic element, so that means it exists in pairs naturally in the atmosphere, right? Kloberfin or Brinkelhoff or whatever it is these days. Whatever. A way to remember all the diatomics. That's right. Cool? So today I want to start by talking about the major events in this respiration system. The first thing that we should mention, at least with respect to glycolysis, is that you start out with a glucose molecule, C6H12O6, and from that you generate two pyruvate molecules. Naming here is important. Two molecules of pyruvate. Now, in this molecule, in glucose, how many carbons do you see? Okay, so you see 6 carbons. You see 2 molecules of pyruvate, and I'll tell you that each pyruvate is 3 carbons. So if each pyruvate is 3 carbons, how many carbons are there total in this system? Okay. So the number of carbons is still maintained. You haven't lost any carbons yet. What have I drawn? Very good. What have I drawn there? Very good, the cristae. Now I've drawn this like with respect to like location to some degree, and you'll notice that I drew out glycolysis and I didn't draw it inside of the mitochondria. So I think that means that we have to be talking about how glycolysis is outside the mitochondria. So I could label this as being in the cytoplasm, which means the event for for the actual process of continuing breakdown would have to be in the mitochondria. So somehow we have to talk about how we're going to get stuff inside. So how are we going to get stuff inside? Okay. Now above, I had mentioned how if you're doing the production of lots of ATP, you're doing it inside the mitochondria, which is going to be aerobic. Which means I kind of have like this division line. And to the left, what does everything have to be? So everything over to the left has to be considered anaerobic. And everything that is post this point is going to be considered aerobic. Which means that it is directly requiring H2O. I'm sorry, not H2O, O2. Where's O2? Whereas anaerobic would be requiring no O2. Good? So obviously the next event is going to be the pyruvates having to go inside of the mitochondria. Now, if you had to guess, what would be the regulation in pyruvate being able to travel inside of the mitochondria if O2 is present? No O2, then you wouldn't continue forward. So somehow, and this is going to come tomorrow, we really have to describe how you could stay out in the cytoplasm, but that means that you would be doing a process called fermentation, which means that you would be anaerobic still. But again, we're going to show, for the sake of showing mass ATP production, we're going to go ahead and continue on inside of the mitochondria. We're going to show aerobic. Now, once inside the mitochondria, it's actually no longer pyruvate, it's a different molecule. But there's still two of them. The name of the molecule is acetyl CoA. The CoA stands for coenzyme A.